Hello boys, welcome back to another session of online NDA UPSC training video. This session we are focusing on biology part of it, and in that particularly cell and cell organelles of class 11th chapter. I have been telling you that this chapter is very important as far as your UPSC-NDA is concerned, because the pet names that are being given to various cell organelles will be asked frequently in UPSC-NDA examination. And I remind you that 40 to 44 marks worth biology questions will come. And in that, 8 marks will be from this chapter on an average. So it is always better to prepare this part of biology so that you will get all the questions correct, and 8 marks at national level would do lot much of difference. So here we go, some questions let us answer. Polyribosomes are aggregates of. Poly, as you know, many. Ribosomes are the cell organelles that are responsible for protein synthesis. In fact, we say ribosomes are protein synthesizing factories. Remember? So the alternatives are polyribosomes are aggregates of. Ribosomes and rRNA, only rRNA, rRNA is ribosomal RNA, peroxisomes, and D, several ribosomes held together by string of mRNA. So particularly computer boys, you know when you are studying biology in class 10th, inside basically one living cell you have to imagine, and inside the living cell there is a liquid called cytoplasm. Inside the liquid cytoplasm, all cell parts, we call it cell organelles, will be floating, and one of the cell organelles is the nucleus. Inside the nucleus, again one more liquid is there, nucleoplasm, and in the nucleoplasm, chromosomes, ribbon-like structures, and inside the chromosome, DNA. Okay, so DNA and RNA are the nucleic acids. They are the secrets of our characters. So in RNA, there will be 3 types. One is called rRNA, ribosomal RNA. Second one is the mRNA, messenger RNA. Third one is tRNA, transfer RNA. So polyribosomes are aggregates of several ribosomes held together by a string, means strip of mRNA, single thread mRNA. Second, plasma membrane is made up of A, proteins and carbohydrates, B, proteins and lipids, C, proteins, lipids as well as carbohydrates. That is the correct answer. D, proteins and some nucleic acids and lipids. Because Singer and Nicholson, 2 scientists, found this fluid mosaic model, and there when we analyze chemically, we find that plasma membrane is made up of proteins, lipids, fats and carbohydrates. Then third question, fluid mosaic model of the cell membrane was put forward by, just now I said, Singer and Nicholson. Question 4. Addition of new cell wall particles among the existing ones is deposition, apposition, intersection and aggregation. Of course it is addition. Addition is always apposition. Cell wall shows complete permeability, semi-permeability, differential permeability, impermeability. What is this? What do you mean by permeability? To permit. Permit something that comes inside and that goes outside. There are certain things that come inside, okay, like water, oxygen, and there are certain things that are to be thrown out of the cell, like all the waste materials. So who gives this permission? That is permeability. Plasma membrane, like our skin, is semi-permeable. It will allow only after screening, whereas the cell wall is having complete permeability. No screening, it just allows. Question 6th. Ribosomes were discovered by, of course, Palade, a scientist by name Palade. And then ribosomes are the centers for C. Just now I have been telling you, protein synthesis. And an outer covering membrane is absent over A. Nucleolus, lysosomes, mitochondria, plastids. Lysosomes, mitochondria, plastids, in fact, they'll have double membranes, outer membrane and inner membrane. Whereas nucleolus will not be having any membrane, it is absent. All plastids, question number 9, all plastids have similar structure because they can store starch, lipids and proteins, get transformed from one type to another, that is the answer, and perform same function, be present together. So it is, they can get plastids. All of you know that plastids are of 3 types. Chloroplasts, which are responsible for green color of the plants, because chloroplasts will have chlorophyll, a green colored pigment. That's why plants are green in color. And chlorophyll not only gives green color to the plants, but also helps in photosynthesis. Remember in your 10th class biology, life process 1 and life process 2, wherein you studied light phase and dark phase, right? And chlorophyll, with its presence, absorbs sunlight, and CO2 plus H2O takes place, formation of starch. Starch is the food of the plant, and we call this process as photosynthesis. But of course, class 11th and 12th biology boys know it, that directly CO2 and H2O will not combine, but they are responsible, the raw materials are responsible and they need to be present for photosynthesis. So plastids are of 3 types. Chloroplasts, as I said, green colored one. Chromoplasts, all colors other than green, red color, brown color, like that. Chromoplasts. And leucoplasts are the colorless ones. But even though they don't have color, they store food. That's why chloroplast. Next, question number 10. Oxysomes or F0, F1 particles occurs in. Of course, when we talk of F0, F1, you know that ATP, adenosine triphosphate, we call it energy currency of the cell. That will be present in inner mitochondrial membrane. So A, thylakoids, no. Second, mitochondrial surface. Third is the inner mitochondrial membrane. In fact, cristae. On cristae, this F0, F1 particles will be there. And chloroplast surface, no. Question 11. Which one is apoptoreticulaire interno? That is the name Golgi apparatus. Okay, Golgi apparatus has got that name. It's a Latin one. And which is correct about cell theory in view of the current status of our knowledge about cell structure? A. It needs modification due to discovery of subcellular structures like chloroplasts and mitochondria. Modified cell theory means that all living beings are composed of cells capable of reproducing. Cell theory does not hold good because all living things do not have cellular organization like viruses. And that is the answer. Cell theory means that all living objects consist of cells, whether or not capable of reproducing. Next, question number 13. In plant cells, peroxisomes are associated with, just now we said, photorespiration, because ATPs are released, adenosine triphosphate. Adenosine triphosphates are chemical form of energy, and that is why we call ATPs are as energy currency of the cell. Like we call Indian currency is rupee, American currency is dollar, like that. Energy currency is ATP. And name of Sleeden and Swan are associated with cell theory. Just now we talked. 15. Membranous bag with hydrolytic enzymes, which is used for controlling intracellular digestion of macromolecules, big molecules, okay? And it is a membrane structure, spherical structure. So A, endoplasmic reticulum, nucleosome, third lysosome, D, phagosome. So answer is lysosome. Again here I must remind you that lysosomes have got a pet name, suicidal bags of the cell. This question repeatedly asked in UPSC-NDA. Okay, lysosomes are suicidal bags of the cell. Why are they called so? They collect all the waste material, come to the surface of the cell. Because they have that hydrolytic enzymes, they blast themselves. Since they are blasting off, killing themselves to throw the waste material, it is something like a suicidal attempt. That's why we say suicidal bags of the cell. 16. Golgi apparatus is absent in, of course, in bacteria and blue-green algae, cyanobacteria, there won't be Golgi apparatus. 17. Inner membrane convolutions of mitochondria are known as, just now I said cristae. Over the cristae, there will be finger-like structures called cristae inside the mitochondria. In fact, inner membrane of the mitochondria will form cristae, and over the cristae there will be F0, F1 particles, and they will possess the ATP. Question 18. Cell organelles having hydrolases or digestive enzymes or lysosomes. Next, question 19. Organelle or organoid involved in genetic engineering is plasmid. In fact, we define plasmid as circular extra chromosomal circular DNA. Extra chromosomal, other than chromosomal. Extra chromosomal circular DNA. Normally DNA is double helix, just like ladder. Nichanage. You hold the ladder and twist it. Two people hold the ladder and twist it. How you get the shape? That will be the shape of DNA. But here it is circular DNA. So that's extra chromosomal circular DNA is called plasmid. Okay? And that is being used in all the experiments in the research work of genetic engineering. And thanks to genetic engineering, we are able to cure many diseases we never thought we can cure. So thanks to that. And lot 20. Mitochondrial cristae are sites of, just now we said, redox reactions, oxidation and reduction reactions. So we can solve some more in our next session.